So the next speaker is Brad Dickerson. He's an associate professor of neurology. Somebody I think he started probably within a year of myself, so we've sort of grown up together, wrote our first grants together, done a little bit of collaboration together. He's well known for his, do you need a plug-in? Okay. For memory analysis, and I'll turn it over to you, Brad.
Do we have to switch to this? I don't know. Oh, it's already there. Great. No, it doesn't matter. Great, thanks, Steve, and it's a real honor to be here. I don't have nearly the level of stories to tell. I don't think I'm humbled by the origins of the NMR Center, but I want to just kind of give you a little bit of my background in coming here and talk more about some of the newer things that I think are we're learning more and more from fMRI. This is, I've been putting this at the beginning of my talks lately because I have a number of patients who themselves or whose family members are artists, and the more that I've been sharing what we learn from their scans with them, the more that some of them are inspired to make art from it. So you might be able to tell, you might be able to identify this little thing as a brain structure. And so these, all of these are, when I sent the scan to this patient's wife, this patient with frontotemporal dementia, she traced the contours of several sections and said, you know, that's my husband's brain. And then she painted her feelings in coming to grips with his diagnosis, which was made so much more vivid to her by seeing the images. I find time and time again that when I'm trying to tell a patient what's wrong with their brain that's leading them to behave in these strange ways or forget things or not be able to find their way, they get it so much more when you can show them the pictures. So, I just think that's one of the most powerful aspects of medicine that I do, is to try to take this abstract dysfunction of the brain and turn it into something concrete that people are interested in finding out, the monitoring of and so forth. They want a follow-up scan to see if their brain has changed. And so it's a lot of fun to use these tools in that way. So I was at University of Illinois, Chicago. My bachelor's is in biomedical engineering, and I went over across the street to Rush Medical Center in working with Leila de Toledo Morel and Frank Morel, and we consulted with Gary Van Heusen in Iowa to do these traces of the hippocampus, and we developed a protocol for the entorhinal cortex. And so I spent a lot of evenings and weekends in medical school tracing serial sections of the entorhinal cortex and the perihippocampal cortex and the gyrus, really, because we were mostly getting the full gray matter and white matter subjacent to it. And while I was doing this, and our applications were both in epilepsy but also in mild cognitive impairment and Alzheimer's disease, Chantal's paper came out, and I said, can't we do fMRI? And it was funny because at Rush we would go after 6 p.m. or on the weekends, and the clinical folks would be clearing out to make room for the research MRI sort of after-hours party. And we would have old people and young people and all variety of control and patient populations coming in after hours on the weekends. But the whole thing was really run very much like a clinical service with the technician there all the time, and so we couldn't touch our hands to the console. And as a biomedical engineer, I said, I really like the anatomy, I like the data analysis, but I'd like to get in there and push some buttons in the console room. So when I came in '98 to do a rotation in neurology at Mass General and took a trip over to the NMR Center at the time, I was so happy to see everybody down there fiddling away with their engineer and other physicist hats on. And so I thought, wow, this is really a community where I could be part of the team that's tweaking things. So that was really the first inspiration that led me to think, if I go to Mass General, or if I can get to Mass General somehow for neurology residency, maybe I can find a way to get in to work with the amazing people at the Martino Center, or what became the Martino Center. So I was really happy to have the fortune to end up there. And so in 2001, I started working with Marilyn Albert, and she said, well, let's take you down and introduce you to some of the people that really are doing things. So she introduced me to Jen Holmes and Julie Bates, and through neurology training, I was already familiar with Vern Cavanis and had met Dave Kennedy. And so, you know, it was really great to see these all variety of people, both senior and junior, that were in there really contributing to the mix of ideas and implementation. And so Chantal showed the image from that first study where we had kind of taken the blobs that were in her first paper and said, well, where are these blobs really? And, you know, can we get to some level of detail in the medial temporal lobe using her paradigm in patients with mild cognitive impairment? And then with Risa Sperling, we did a similar analysis with the face name memory paradigm and found this increased activation that was at the time thought to be maybe an artifact. And you know, I went through every individual patient and said, you know, this guy's got a little bit of atrophy. You know, I was blind to their clinical status. This guy's got a lot of atrophy. These each are the median members of these groups. And I said, you know, this patient looks like they have AD based on their atrophy pattern, and there's really no activation to speak of. So I think the group data probably makes sense. And one of the things I really loved from the beginning was FS-FAST's get under the hood and see the real data and understand what your maps mean. And we didn't even really use maps in these first papers to much degree. I think we really wanted to know what the signal in the voxels meant. So then we started looking beyond the medial temporal lobe and sort of feeling annoyed about these areas of deactivation that we thought, you know, probably should just be ignored as a tail of the distribution that would, you know, help us in our statistics if we could just forget about it. Until the work that we've heard about with resting state data that really brought me back to Gary Van Heusen's teaching. This is his 1972 science paper showing the connectivity of the medial temporal lobe. And we used that background as an inspiration to try to look at the resting state correlations between some of these areas that do different things during memory encoding, and show with Risa again that people that perform better, and these happen to be older people, were the people with the stronger resting state correlations between some of these areas that would fractionate in their activation profiles during the task itself. But this was collected before the task, so really would predict how well they were going to perform on the task. And then I think, you know, none of this would be possible without the amazing neuroanatomical reference space that Anders and Bruce and others created with FreeSurf for itself. And I think that has been one of the real foundations upon which we can build to try to relate the multimodalities of data that Steve was talking about to each other. So this is just an example of a modern lesion analysis that we did a few years ago. It used to be said, and still is sometimes, that Alzheimer's patients have diffuse atrophy. Just diffuse. You know, you can't make anything out of it. There's no lesion analysis you could do. There's no focal lesion. But actually, if you look at cortical thickness and think about atrophy that's beyond what you would see in any normative distribution, you can think about that as a distributed multifocal lesion. So to test that idea, we took a test like the California Verbal Learning Test. It's called the Ray Auditory Verbal Learning Test, where you are first presented with 15 words. So if I read to you a series of 15 words and said, now repeat as many of those back to me as you could, that's really an auditory verbal working memory test. There's no long-term memory involved in that. And so you might predict that if you took a bunch of Alzheimer patients, and this is from ADNI data, and said, well, where are the people who have the worst performance on that task, that part of the task, where do they have the most atrophy? Then you see what's an amazing left lateralized, frontal parietal, and temporal map of what looks a lot like the auditory verbal working memory circuitry. But this is structural data. So it's amazing when you line this up with functional maps how specific some of the atrophy patterns can be when you think about how they relate to specific facets of behavior. And then if you say, now, what about after 5 times when you've repeated that list over and over and over again, and it starts to get into long-term memory systems and begin to be stored, how does that look? And so these are the same patients, just a different component of that task. And now we've got a much more bilateral pattern with the medial temporal lobe present. So the people with the greatest atrophy in these regions are the people that are doing the best on the last encoding trial of this repetitive word learning test. And then you can say, well, what happens with retention? So if you wait 20 minutes and say, now what % of the words that you originally learned, controlling for the initial learning, because all these patients are impaired on everything really, what % can you freely recall? Then it turns out now we've got a very medial temporal, bilateral, paralimbic, posterior cingulate precuneus, sort of temporal limbic pattern that's quite different than encoding. And then finally, for the recollection versus familiarity component, we looked at recognition memory for the items that were not recalled, with the idea that that's familiarity, which you'd predict is perirhinal cortex. And this is a whole cortex analysis. And I said, you know, I'm not sure I really believe in the dual process of recognition memory yet. I haven't really seen a great dissociation in patients, but we ran the whole cortex map, and to me, you don't need statistics to, if you go into this with a hypothesis, you don't need statistics to convince yourself that this is a real effect. It's so localized and predicted by that model that I became a believer after I saw this, and this is a fairly liberal threshold. So we also, along the same time, started making these maps of, on average, if you take a bunch of Alzheimer patients and just say, let's see, as Chantal was saying, sort of the signature of some of these abnormalities. This is the signature with Randy Buckner's data that we worked with of the average pattern of where people have atrophy when they have Alzheimer's. And we were able to take this statistical map and, through the beauties of the spherical registration, make a set of regions of interest that then could be applied to new patients' brains after they're registered through the recon procedure. And then you can use that as a biomarker for a variety of other things. So one thing we did was we said, you know, if the hippocampus is hyperactivating, and that's not just a compensatory effect, if actually maybe it's a part of the pathology and it's maybe pathological hyperactivation, then you'd think the people with the greater degree of atrophy in these structures with presumably worse pathology should have more hyperactivation. And that's what we found when we put this together with this AD signature of cortical thickness. So the lower you are on the y-axis, the more atrophy you have in these Alzheimer's vulnerable brain regions. And within a group of mild cognitive impairment patients, the more that they have that atrophy, and they're not yet demented, they're still able to do the task, the higher their activation. So really supporting the idea that this was the hyperactivation was occurring in the context of a neurodegenerative process and was in some way linked to that process. And then because Marilyn Albert had been following these people over many years, we were able to say after 5 years, what happens to these people with mild cognitive impairment that were scanned back in, at this point it was in 99, 2001 time frame when they were first scanning MCI patients. And so these were 25 people, and over 5 years, they kind of have a pretty heterogeneous course. Some of them don't change very much. Some of them get a lot worse. And so if you say, you know, I would predict that the medial temporal lobe hyperactivation in these people is a reflection of how aggressive or severe their pathology is, then you'd expect the hyperactivation would predict a greater rate of decline over time, and that's exactly what we found. So the more the hyperactivation in these mild cognitive impairment patients, the faster their decline over 5 years. And this was really all made possible because the infrastructure was in place to do this kind of work, and the data had largely already been collected, which I think is so much of what we all, when we're growing up around the Martino Center and MGH in general, benefit from, is this kind of infrastructure with PIs that are doing these ongoing projects that we can plug into and do way more than we would ever be able to do in a single lab. And this now has been translated into further work that is part of a clinical trial. So not one that we're doing at the moment, but planning to do. But there is a group at Hopkins where Marilyn Albert is now who's giving levetiracetam, an anticonvulsant, to patients with mild cognitive impairment to see if you can dampen down that medial temporal lobe hyperactivation just enough. Maybe you can help slow down their progression, which would be a direct hypothesis from that natural history data that I just presented. So I want to just switch gears a little bit and say with the language system, just coming back to some of what Steve was, is somebody going to give me a 5-minute warning, by the way, or is it, okay, about 5 minutes now? Okay, great. So this is a patient with primary progressive aphasia. You would imagine that just based on this atrophy, this image showing this focal atrophy in the anterior temporal lobe. And we did an analysis thinking now more about the network neurodegeneration hypothesis. So within neurodegenerative diseases, there's this idea now that has been gaining support that neurodegenerative diseases, the way they affect the brain is sculpted by the brain's normal networks, and the way they progress throughout the brain and damage the brain follows connectional properties. And so here, this is a little complicated, so we took the hot spot, the area of greatest atrophy in these semantic dementia patients, which is right at the tip of the temporal pole, and we said, show us where there's functional connectivity and resting state with that region. And here's that map, and here's the atrophy map from a pooled sample of 2 different groups of semantic dementia patients, and here's the overlap. And so you can see there's a lot of overlap between the connectivity of the temporal pole in normal people and the atrophy pattern throughout the cortex in patients. So further circumstantial evidence supporting this idea. But what no one had done before was something that Jessica Collins in my lab said, we should try to do this, which is, if you look at any resting state maps, there's some areas that are more strongly correlated and some areas that are more weakly correlated, and we never really know exactly where the threshold should be, but there's a range of strength of connectivity. And you can see similarly a range of atrophy. So if you just say, well, if the disease is progressing through the connections of the network, shouldn't the areas with the strongest connections based on just simple R-value, transform correlation coefficients from resting state data, shouldn't those be the ones that have the greatest relative atrophy if it progresses in a linear fashion, which is a big question, but this is sort of a first-level hypothesis. So I was surprised to see this worked out well and replicated across the 2 samples, where if you take a bunch of healthy controls and you just say, show me the regions, and this is all the regions throughout the cortical parcellation that have any signal in them, so we're getting rid of regions that don't have any signal. All the regions with signal, regardless of how strong that signal is, are included here. And we just say, the areas where there's stronger connectivity are correlated with the areas where there's a greater degree of atrophy. So more atrophy up here, stronger connectivity. So now we're trying to do this same kind of work in longitudinal data, but I think it really provides further support. And one of the ideas that I am sort of kicking around is, could there be individual differences in this? So, if one person, as we've seen from Alex Torotoglo's work and others, has stronger connectivity within a particular network than another person, would they potentially be more likely to have that network be resilient or maybe be vulnerable? So I think these are some questions that we can ask now with a lot of the data that are being collected. And so this is just an overall atrophy map of patients with primary progressive aphasia, again showing the specificity of the atrophy to the language network. So if you just take this atrophy map and make some sort of regions of interest that are canonical nodes in the large-scale language networks, and do functional connectivity maps in normal people, you see something that's kind of interesting, I think, which is that if you put a resting state seed in the Broca's area, you get this map. In the caudal middle frontal gyrus, you get this map. In the supramarginal gyrus, you get this map. More or less similar maps of sort of the dorsal language network, whereas if you put one in the temporal pole, you really get a very different map, suggestive more of the ventral language network that Richard Weiss and others have talked about. If you subscribe to the Hickok and Popple model of language network localization, the middle temporal gyrus is a key interface between these 2 networks. And so if you put a seed in that area based on the atrophy maps in patients and say, where's the resting state connectivity, you see this sort of bridge between the 2 networks that I think really supports that hypothetical model. And if you look at that in relation to task-related activation, so this is the resting state map, task-related map, and the atrophy map. It really is hard to, not be kind of blown away by the specificity of the degeneration to this particular network in these patients. And I'll just close in the last bit by talking briefly about the new tau PET efforts that Keith Johnson and George El Fakhri and Neil Vastev and others have been pioneering at both the main campus and the Martino Center. And so this is Keith's first map of the localization of tau signal from T807 ligands, the T807 ligand in patients with Alzheimer's dementia. This is an Alzheimer's dementia patient. This is a milder patient. These are more kind of MCI patients, and you can see that it follows more or less the pattern that the Brocs have predicted, if you follow that literature, and where tau pathology should be in the course of Alzheimer's disease. This is a single patient's brain showing the localization of tau PET signal based on these methods. And I think we'll begin to see, I think as Steve was mentioning, the amyloid PET has now penetrated the clinic, and I think it won't be that long before we start to see tau PET penetrate the clinic as well and potentially be usable. But it seems more complicated so far, so I think it might take some time still. But this is a map of one of the Alzheimer patients, and this is a map of a patient with a non-fluent form of progressive aphasia, and I think you can really see the difference being the lateralization of tau PET signal in the context of this patient's atrophy. And if you make a surface projection of that, you really again see this remarkable concordance between where that patient's atrophy, atrophy is, and that's the atrophy map, and their presumed tau PET signal. And I say presumed because there's still a lot of validation work that needs to be done. But if you then look at that in the context of the networks, you see again that the network structure of the language network corresponds beautifully well to where the presumed tau PET signal is, and the thickness is maybe lagging behind a little bit, suggesting that may be the way it progresses. And this is a longitudinal scan, again, supporting the idea that if we think about networks from the perspective of how we define them with fMRI or maybe diffusion, tensor imaging, we can make clear predictions about where we should see these other modalities of signal like tau PET signal. And so I think if you go into it like that, then this is really remarkable in the sense that at baseline, this patient had frontal and temporal elevated signal and a little bit of posterior temporal signal. And 1 year later, the focal signal that began is higher, but there are also areas that could look sort of almost like they're below the threshold, like there's a little signal right there that emerges a year later as more prominent. So again, I think really supporting the idea that if we understand the connectional properties using fMRI and similar mechanisms of the brain, we'll be in a better position to predict what we should see with not only atrophy maps, but also with new modalities of data like tau PET. Thank you very much for your attention, and thanks to Bruce and everyone for putting this together.